myself included, cDNA or complementary DNA, an apology, because we often use the terms gene or mRNA when really what we're talking about is the complementary DNA, which is a DNA version of mRNA. And mRNA is messenger RNA, and it's an RNA copy of a DNA gene. So a DNA gene has the instructions for making a protein. A messenger RNA copy of this gene is made, and this copy is not identical to that gene because this mRNA has the regulatory information, the introns, taken out. So the mRNA represents like an edited version of this gene, but that editing is happening at the RNA level, so it's not messing with that original gene. So you still have all that regulatory information in the nucleus and stuff where the DNA needs it. But this messenger RNA, it doesn't have those regions that the nucleus needs, but the ribosomes, the protein-making machinery, don't need. And the ribosomes wouldn't know what to do with that regulatory information. And similarly, if you were to stick the DNA, if you were to stick the gene for making a protein into something like bacteria, bacteria wouldn't know what to do with all those introns. It's not going to be able to process it. And so if we want to get bacteria to make a protein for us, we want to stick in the cDNA rather than the gene. But often we just talk about like cloning a gene because it's a lot simpler, and especially if you're talking to people who aren't familiar with molecular cloning and these sorts of things, it's easier to just say gene. But really what we're dealing with is the cDNA. We're often also dealing with the cDNA when we're doing something like RNA sequencing or qPCR to measure how many copies of a messenger RNA there are, or to see which messenger RNAs are present or at what levels. And so basically the cDNA is going to be more stable than the RNA. So RNA is very fragile and it's sensitive to like RNases and things that are all around and can chew up RNA. And so if we can convert it into cDNA, well, now we have a more stable source and we can more easily manipulate it in the lab using techniques like PCR, which can make DNA copies of DNA, but they don't make DNA copies of RNA. To make DNA copies of RNA, we're actually going to use a reverse transcriptase. And I'll tell you more about this as well as different strategies for making cDNA with different types of primers and things like this. Once you have that cDNA, you have that DNA copy, you can do like a first strand, make one copy of it, one strand of a copy, then you can use that as a template for making a second strand. Now you have double-stranded DNA, you can do things like stick it into plasmids, make cDNA libraries representing all the different mRNAs that were expressed in a given sample. You can then probe this, you can fish out the mRNAs for various proteins. So some of these techniques that are used with these cDNA libraries have been like superseded by more recent methods now that we have all these like whole genome sequencing and things like this, but there's still species and samples where you don't have all that information, and so this provides a way you can fish out that sequence, and it also provides a way if you can't find the cDNA you're looking for in some sort of library like AdGene or DNASU, and you want to work with that gene, or I mean with that cDNA, not the gene, because the gene would have all the introns and stuff, whereas the cDNA, this is just representing the transcripts, the messenger RNAs. So a cDNA library is going to be a representation of the transcriptome, or all the transcripts present in a sample. And this is going to vary from cell to cell, so you can do things also like subtractive hybridization and things to figure out what is different between different samples, which cDNAs are expressed in some cells but not in others, and various things like this. And so this has played key roles in determining, like, in solving medical mysteries and things, especially historically. So lots of cool stuff that you can do with cDNA, but you also need to be careful with cDNA because you can also have alternative splicing where you can get multiple cDNAs from the same gene, thanks to those introns being able to be removed in different ways. And so if you want to, say, get a bacteria to make a protein for you, you want to make sure that you're having them make the version that you want them to make. And so you want to make sure that you have the cDNA for the right splice isoform you want, and you also need to make sure that you have a complete cDNA, because basically when you make that cDNA, it not necessarily have the whole full-length cDNA, and so you have to be careful and make sure that you get the full-length thing. So we'll go into a little more of these details as well as how we can find some of these sequences and things like this, some terms and various other stuff like that. So let's go on. When your cell goes to make a protein, the basic overview of how it gets made is first it takes this original recipe, the gene, which is written in double-stranded DNA. It makes an RNA copy of it. So this original RNA copy may copy word for word, letter for letter in the process of transcription. But this original immature messenger RNA, this transcript that you get here is going to have this regulatory information that was present in the gene in the form of these introns, which are kind of interrupting regions between the exons, which are the regions that actually have the instructions for making a protein. And so this RNA copy needs to get edited, and those introns need to get removed. These introns can get removed in different ways to give you alternative splice isoforms. So I have much more on that in another post, but the basic reason why I'm bringing it up here is because you can get multiple different messenger RNAs from the same original gene. And so by splicing it in different ways. And this is going to give rise to different cDNAs representing different splice isoforms. And so you might see different mRNAs from the same gene, both within a sample as well as between samples, because cells can make and regulate differently these different versions, these different splice isoforms. And so that's something you're going to have to keep an eye out for when you are if you're trying to do something like clone a cDNA, get cells to make a protein, you need to make sure you know which isoform you're working with, and we'll get into that. Speaking of getting that cDNA, basically the cDNA stands for complementary DNA, and it's a complementary version of the messenger RNA. So we use this term complements when basically the DNA and RNA letters, as well as DNA and DNA letters and RNA letters, they have bases. These are like these letters, they have these parts like those A, T, C, G, U. Each of those is going to have a complementary base where it's going to form this one-to-one pairing, where this allows us to use one strand as a template for making a copy of the same strand. And in our cells, we have a couple ways this happens. We have a DNA-dependent DNA polymerase, which uses DNA to make a copy of DNA. This is what our cells do when we want to copy all of our DNA before dividing. And it's also what we can do in the lab when we want to make copies of DNA with a technique called PCR. And so this is going to be one of the reasons why cDNA is so helpful, because PCR is so helpful. We can make lots and lots of copies of a region of DNA using this technique. But we can't make lots and lots of copies of DNA from RNA using this technique. Instead, we need to make DNA from that RNA first. And this is what we're going to be doing in this reverse transcription step. And this reverse transcription step is not something that we actually do in our bodies, but some viruses do. They have this reverse transcriptase, and we can take advantage of the enzymes of the molecules, the molecular-like machinery that it uses in order to make these copies, and we can do it in a test tube. And this is going to allow us to make that cDNA. It's called reverse transcription because transcription, as we had talked about before, was that process of making the RNA copy from DNA. And so reverse transcription is making a DNA copy from RNA. And we can make this DNA copy of the RNA, and we call the cDNA. So unlike the gene, the cDNA doesn't have the introns. It's going to be the edited version. But unlike the mRNA, it's going to be more stable, and it's going to be easier to genetically manipulate. And so the cDNA is going to be really useful for various things that we'll get into, including seeing what versions of proteins are being made through techniques like RNA sequencing, qPCR, northern blot. And it's going to allow us to get cells to make proteins of interest by basically cloning that cDNA into plasmids, getting bacteria to make it through recombinant expression and things like this. So that cDNA is going to be really, really helpful. So how do we actually get it? So as I mentioned, making the DNA from the RNA is a process called reverse transcription. And there are various viral reverse transcriptases that are used, such as like AMV and MLV or something like that. But these basic reverse transcriptases that can be used in the lab. But they need to have a starting point. So like normal PCR, where basically you have a primer, you have to tell this polymerase. So whether it's a reverse transcriptase or whether it's like a DNA-dependent DNA polymerase, basically one of these enzymes is going to put together these DNA letters. It needs to know where to start. And so we specify where to start using primers, which are these short DNA sequences that bookend the region that you want copied when we're doing things with PCR. The first thing what we're doing when we're generating cDNA is not PCR. What we're doing is we're doing first strand synthesis. So basically we want to make a complementary version of that messenger RNA. And so we're going to be making a copy of the, replica of the template strand. But we need to tell it where to start. So we're just going to have a single primer, or at least, we're just going to have one end of the primer. You'll see what I mean in a second, hopefully. But we're not going to be bookending a region. We're just going to have one side of it. And so how are we going to capture all of the mRNAs? Well, one feature that mRNAs have in common is that they have a 3' poly A tail. So when I talked about the processing of messenger RNAs, I talked about splicing, where we remove those introns. But that's not all that happens. It also gets a 5-methyl 7-guanosine cap and a 3' poly A tail. And so this poly A tail is a bunch of A. And what matches with A? Well, T matches with A. And so we can use a poly T primer, a LIGO DT primer. So a LIGO basically means short. DT, this is a deoxythymidine. And basically it's going to have a sequence of T's. This is going to bind to that poly A sequence, and therefore it's going to capture these transcripts because they have this poly A tail. But there's some problems with this. They can also make copies of parts of RNAs that just happen to have a lot of A's somewhere. And it can start anywhere in the tail. And so some of these RNAs might have really, really long tails. And therefore they might have this primer bind like at the end of the tail. And now the polymerase is going to run out of steam before it even gets to the gene. One way that this can be avoided is using like anchored oligo-DTs. So at the end, they have a non-T, so they only start at the beginning of the tail. But if you basically, this stuff works when you're just trying to get the transcriptome. So when you're just trying to get the cDNA, the messenger RNA, the cDNA from the messenger RNA. So you just want like the express things, your transcriptome, because we call it like the messenger RNA's transcripts. So we can refer to the cDNA compilation as like the transcriptome, a representation of the transcriptome. But basically, this isn't going to work if you wanted to look at things like non-coding RNA. And it's going to give you a bias towards the 3' end. So even if you have one of those anchored oligo-DTs, you're still going to be starting at the 3' end. And if you have a really, really long messenger RNA, you might not get to the 5' end. So for various reasons, you might want to have a primer that's actually going to bind internally inside your messenger RNA. If you know what specific messenger RNA you're looking for, you can define like sequence-specific primers. But if you don't, you can use like random primers if you want just like a general representation of all of the messenger RNAs. You can use random primers that basically have a mix of random letters that are bound to bind somewhere. Of course, when you're doing something like this, then you're not going to get the full-length cDNA. Whereas if you go from the tail, then you're going to hopefully get the full length, but you might miss the 5' end. So if you're trying to find a sequence based on a cDNA, you might actually need to combine overlapping sequences. And I'll talk more about this, more about this a little later. But somehow you can generate that first strand. And now when you have that strand, you have a single-stranded DNA. You can work directly with a single-stranded DNA for some purposes, and then for other purposes, you're going to want to do a second strand synthesis where you use that first strand of DNA as a template for making a second strand, and then you can stick that in plasmids and things like this. But just having that first strand is going to allow us to do things like use qPCR to search for specific sequence. So qPCR stands for quantitative PCR, and often we use the term qPCR when really we're talking about RT-qPCR or reverse transcription qPCR.
But sometimes the RT also stands for real time because you're measuring it in real time. So you might see like RT, qPCR. The acronyms can get, or abbreviations can, whatever, can get really complicated. But basically the idea is that you're making copies of specific messenger RNA, and then you're counting those copies as they get made, such as by using a double-stranded DNA binding dye or a reporter probe that's going to bind specifically to the transcript of interest. And so in this process, basically, the more copies you start with each cycle of the PCR, you're going to double those copies. And so the more copies that you start with, the quicker you're going to have a buildup of these copies that you can then be measuring. And so the more copies of a transcript that are present in a sample, the more that messenger RNA is being made, the more theoretically of the protein that's getting made, the stronger the signal that you're going to see, and then you can measure this. And so this is a technique that's often used as one of the measures of like gene expression. And so when we say gene expression, we can be meaning different things. But a lot of times when we're talking about gene expression, we're talking about the making of proteins. And so basically, if you want to know about the making of proteins, well, you can look at how many recipes that are made. And this would be the idea with your RT-qPCR. Another related technique is going to be a northern blot. You can also do this with your single-stranded RNA. qPCR has become more common, kind of superseded the northern blot in a lot of ways, but northern blotting is still really useful for various purposes. If you're looking for, say, for modifications to RNA and things like this. And basically you use probes, these labeled sequences to probe a membrane for various sequences. So if you know about a Western blot looking for proteins, a Northern blot is similar in concept, except that you're looking for RNAs with DNA probes. So those were some things that we could do with that first strand, but for some things, we're going to need the second strand as well. There are various strategies for getting the second strand. Some of them involve like using like an RNase H, so an enzyme that's going to cut RNA DNA hybrid. It's going to cut the RNA, and if it kind of like nicks it, it can make starting points for DNA polymerase to generate the second strand. There are all the like strand switching things. There are various different methods, and so I'm not going to try to go into them all. But basically, somehow you're able to use that cDNA strand that you generated as a template for making the second strand. And once you have the second strand, well, now you can do things like you can sequence this. So like RNA sequencing, you can stick like adapters on the end of it that are going to allow it to be sequenced. And so different things like this. There are also a few terms that you need to make sure you don't get confused by. So basically, when the ribosome is reading the RNA instructions, it's reading it in these codons. So like 3 letters by 3 letters by 3 letters. And the genetic code is basically not overlapping. And so where it starts reading is going to dictate what reading frame it's in. And it's going to know where to start because there's going to be a start codon. So in AUG, it's going to tell it, okay, start here. And then it's going to go until it reaches a stop codon. And when it reaches one of the stop codons, it's going to know when to stop. And so in between that start and the stop, you have what we refer to as an open reading frame. And if you use a tool like xParam and stick in a sequence, it'll show you all these open reading frames. So basically it goes from a start codon to a stop codon. So if you see the term open reading frame or ORF, that's representing the start codon to the stop codon. And there can be multiple open reading frames, but typically only one of them is going to be actually the right one. So ORF is one term that you might see as a term associated kind of with cDNAs, because that cDNA is likely has the ORF. It should have the ORF that you're looking for. Another term you might see is going to be EST. EST stands for Express Sequence Tag. It's basically like a short sequence from a cDNA library, like a short sequencing read. So it's not like the full length thing. This was used a lot more in the past when technology wasn't as great, and so you were only getting these short reads. So you might see the term EST. Also in those cDNA libraries, you might not have the full cDNA because for reasons like not making it all the way to the 5' end, or for if you're using an internal primer. So you might need to kind of like overlap, use one piece to kind of fish out the next piece and kind of get the sequence together that way. So various complicating factors, but cDNA is very, very helpful. Just make sure you know what you're working with. You can also stick it into plasmid, so like circular pieces of DNA to make proteins from. And so this is where we can make recombinant proteins. So we're recombining that cDNA that we made or that we got and recombining it with this plasmid, this vector plasmid. So basically the circular piece of DNA that we can put in bacteria, and then the bacteria will make it for us. And going back to what I was talking about before, where there being alternative splice variants of the same gene, basically, if you stick different cDNAs into the plasmid, you can get the cells to make different versions of the protein. But it's important that you know what sequence that you're putting in. And it's important that you put in cDNA and not the original gene because the bacteria wouldn't be able to do that splicing. You can also take like messenger RNA. And so basically using one of those techniques like the poly-A primers or things like this, we can generate cDNAs of all the mRNAs that were present in a sample. And then we can take those cDNAs and we can actually stick them into plasmids, not for expressing, not for making a protein, but just for making lots of copies of those messenger RNAs and kind of making a stable version of them. Because remember, RNA is really fragile. If we want to have a kind of like a long-term way to store these things, we wouldn't want to store it in RNA, and we want a version that we can make lots of copies of. And so it turns out that we can do this, make these cDNA libraries, we can stick these into these vectors that are just going to be hosted by bacteria. So bacteria are basically going to hold on to them and they're going to make copies of it for us, which is really, really helpful. And so, but we need to then, like, we don't even know what's in that library. It's just a bunch of different sequences. And so each of these plasmids is going to have a different sequence in it. But what we can do is we can actually probe that library using like labeled probes that we know like a sequence, we know part of the sequence, we can then fish out the whole sequence. And this is helpful in solving, this is sometimes was used classically to do a lot of things like find disease genes, or not genes, but see which cell types these disease genes were being expressed in. So more on this in my post on cystic fibrosis. But basically they were able to find a gene with a mutation in it. They were able to take the sequence from that gene and use it as a probe for probing for the cDNA. And they did this in different tissues, and they showed that they were able to see this was like highly expressed. So there was a lot of the messenger RNA in the tissues that were most affected by the disease. So this really helped tie together the gene and the mutation and the disease. In addition to identifying whether a transcript is present in a sample, which again, these days is typically done using like a qPCR, but basically you can also do other things, like you can fish out the whole sequence in order to subclone it, so transfer it into another vector, such as one of those expression vectors for making a protein. So you might take a cDNA library, fish out a sequence of interest, the mRNA of interest, then stick it into a plasmid, stick that plasmid into bacteria and get bacteria to make the protein for you. These days, it's a lot easier to, if possible, order the plasmid that you want through a repository like AdGene or DNASU. But you might be restricted because they might not have like all those different splice isoforms and things like this. And they don't even make it that easy really to figure out which splice isoform you're actually working with. For example, in my PhD work in grad school, one of the proteins I was working with was this casein kinase 1A, so CK1 alpha. And this alpha, basically the CK1 alpha, it itself had multiple versions. It had splice isoforms. So if I look up this in UniProt, which is basically like the go-to place if you want to find out about a protein, if you go to sequence and isoforms, you can see that there are 3 isoforms produced by alternative splicing. And so one of them is going to be dedicated as like the canonical sequence, and then there are going to be others. So sometimes the canonical sequence is the most common, but not always. And basically then there are other versions, and you can see how they differ from the canonical. So they might have be missing a sequence, maybe like a localization sequence, or something like this. So basically you can see that each of these is going to have this associated like protein number. And then you're also going to have, if you go down to the sequence databases, you'll have these links to the cCDS, which is going to be kind of like it's going to have an annotated source of agreed upon consensus cDNA sequences of like the full length version for a different protein. Because you'll see, like, based on what we talked about before, you could have all these different cDNAs from the same transcript. Some might be partial, some might be full, some might be like have errors in them. But people wanted to say, okay, well, what's the consensus sequence? And so they generated this cCDS database. And you can see that you're going to have one of these entries for each of the different protein isoforms that you have here. And so if you were to click on that, well, now you can come and you can see the nucleotide sequence as well as the translation. And the blue, where it goes from blue to black, this is indicating that there's like, it indicates alternating exons. And then red highlights the amino acids that are encoded across the splice junction. And so you can see that you're going to see different sequences for each of these different things, and it'll show you both the nucleotide sequence and the translation. So if you have this will tell you the sequence that you would want if you were to go and make the gene thing, or if you were to try to look for a plasmid with it. So if you were to go and you could search for CK1 alpha in like add gene, you can find this plasmid, and it's not even going to tell you what isoform it is. But if you go to the sequences, well, now you can see the sequence up here, and then you can go in and you can search that against the isoforms. It might be easiest to actually just take this sequence and stick it in XPASI prop param. This is going to tell you, basically, it's going to translate it, or XPASI translate. You can stick this sequence in, figure out what the RNA sequence or what the protein sequence that it's in. So see, it's showing you the different open reading frames, and this is going to be the one that we want. And then we can compare those to the different isoforms to see which isoform it actually is. So I don't know of any like really easy way to do this, but this is going to be a way that's going to work. And so I can see that it's like this transcript here, this isoform 1. Yeah, so basically you just have to be careful. And if you know which isoform you want, make sure that you find that right isoform. And if you don't know, make sure you don't care, like just make sure you know what you're working with. Also, be sure that once you actually get this plasmid, then you're going to go sequence it yourself. And be sure that you know what you're working with. So basically this, when you get a cDNA, it's often in some sort of donor plasmid. That's basically just this generic plasmid that's really great for holding DNA, but not for expressing it or anything. So you can see that this basically doesn't even have a stop codon. And so this ORP has no stop codon, and this is going to basically, in order to actually make a protein from it, you need to have it have a stop codon. And so you'd want to stick this into a plasmid, but leave the stop code, have the stop codon encoded by that plasmid. So insert it so that there's a stop codon then put onto the end. And then you could transfer this into different vectors. But you can often find the cDNA you want in something like AdGene or DNA SU. And then alternatively, you can do something like try to fish it out of a cDNA library. And yeah, so basically you can also like order a whole cDNA synthesized depending on what's cheapest and that sort of thing, how much time you have, lots of different options. But hopefully this helped you understand things a little better.